can you check the YouTube, please? Or
Yeah, I'm admitting people. If somebody can help me.
Oh no, for the question part.
Okay, let me see.
Or I can do it later on. Can you check the? Okay. So I think we are live. So welcome everyone to the last seminar of the season. I'm going to get started by sharing my screen. If I can find now share the screen option here. Okay, here we go. No. Where is this? Sorry, we have enough experience doing this and then it doesn't work. Yes. I decided to do a whiteboard. Okay. Good. Okay. So this is our last seminar of the season. But before we get started, I will actually like to thank our previous speaker. I don't know what's going on. Well, yes, there we go. Our previous speaker, Amia Ilani from Bar-Ilan University, for a great talk on higher access. Mostly, I think we learned a lot about this species and a lot about social behavior and social networks, which is also a topic that we're going to be chatting about today. But before we go into our talk, I just want to sort of let you know a little bit about our program for next season. We are already preparing and working on awesome talks for the season number 9, which is also the 4th year of the FINE series. So we're welcoming everyone to come and join us on the 3rd of September, which is going to be our first meeting. We'll be in touch telling you what the plans are for this day, but we also welcome you to join every talk that we're planning for you. So you see, there's not everyone yet there, but we hope to have this ready and then share it with you widely through our email list. Today, our talk is going to be, our presenter is Sonja Wild, Dr. Sonja Wild. I met her at the Max Planck Institute of Animal Behavior, where she was a postdoc, and I was a postdoc as well. She was in Aplin's Lucy's lab. Lucy also had a fine seminar here in the series. So Sonja got her PhD at University of Leeds in 2019, so she's very early career. At this moment, she's a postdoctoral fellow with a fellowship from Switzerland, right? And she's based in University of California, working with Andy C. and Jen Smith. Both of them have given fine seminars here as well, which is really good, a very nice community. Well, Sonja's interests are basically understanding the mechanisms and their underlying behavioral plasticity and she focuses on social animals, as you will see in her presentation. But she is really keen on understanding how animals acquire behaviors through learning and basically how external and internal factors influence these decision makings as they engage in social interactions with other animals. Well, Sonja has a great publishing record, even though she's really early in her career. She's been publishing widely in Current Biology, Journal of Animal Ecology, Philosophical Transactions, Biology Letters, Behavior, and so on. She's very behavioral ecologist. And she also engaged in media. So she has a really good media cover of her papers, and a really good communicator in science, very passionate about nature and photography. If you go on her website, you can see like a really cool photo shots that she has for animal and nature. Well, with that, Sonja will present on when, who, and what to copy, investigating social learning strategies in wild animals. And I just want to thank you, Sonia, for accepting our invitation. And from now on, we will love to hear your talk.
Thank you so much, Adriana, for this very nice introduction. I am delighted to be giving today's seminar. So thank you so much for inviting me. All right, just getting set up here. Wonderful. Okay. My research mainly revolves around the mechanisms that lead to behavioral variation, as Adriana has already mentioned. And this includes both the mechanisms through which animals acquire behavioral traits, but then also once they are knowledgeable, how do they make decisions on which behaviors to express depending on the environmental cues they receive. And I'm interested in how these processes then shape the behavioral diversity but that we see on higher levels of organizations, such as the population or the social group, and ultimately how that affects fitness of both the individuals and populations as a whole. And we are going to briefly touch on all of these topics today, although a large focus is going to be on that first box, namely how animals acquire their behavioral repertoire in the first place. And throughout my career, I have focused my research on wild animal populations, which of course comes with certain logistic challenges, but it also really gives us invaluable insights into like how acquisition of behavior and decision-making processes happen in natural settings within animals' natural, social, and physical environment. I have, during my master's and PhD, I have worked with bottlenose dolphins in Australia, have then moved on to great tits for a first postdoc in Germany. And since 2022, I am now working with California ground squirrels. And given the time constraints, I will be focusing today's seminar on the work I've been doing on the dolphins and great tits, but I'm definitely more than happy to chat about some of the questions I am working on at the moment in the ground squirrel system, if anybody's interested. Now, how do animals acquire behavior? How do they go from being completely naive to knowledgeable? We distinguish between three main drivers that can influence the acquisition of behavior. First of all, many behaviors have some sort of innate basis, with certain aspects of behavior being genetically determined. Take some of the nest-building birds, for example. Even if they're raised in complete isolation, they still have some innate knowledge that for reproduction, they need to build a nest. They might not know how exactly, but some of that behavior is genetically encoded. Second, we have environmental factors that can influence what behaviors are acquired and expressed. For example, the availability of tools in one place versus their absence in another place can determine where we might observe a certain tool using behavior. And finally, as a third driver, we have learning processes that determine what behaviors are acquired and expressed. Learning can entail two forms: asocial learning that occurs through direct sampling of the environment. It generates information that is up to date and adapted to the current conditions. It can also be costly if, for example, it requires a lot of trial and error learning. On the other hand, we have social learning, which is essentially scrounging on information that others have produced. So it's potentially a very rapid and safe way to acquire novel behavior or information. But there is the risk of copying potentially outdated or even maladaptive behavior. So to maximize the benefits of both asocial and social learning, individuals need to employ social learning strategies by deciding when, who, and what is beneficial to copy. And during today's talk, we're going to focus a lot on some of the who strategies in relation to, particularly to generational learning. We distinguish between different forms of learning depending on whether it occurs within or between generation. This first one, vertical learning, refers to learning that specifically occurs between parents and their offspring. Oblique learning also occurs from older to younger generations more generally, so not necessarily parental individuals. And horizontal learning is learning that occurs within generations, so among similarly aged individuals. And the extent to which individuals engage in any of these strategies is heavily influenced by a species' social system, because social transmission of behavior or information can only really occur among individuals that also spend time with each other, so that are associated. But we will circle back to this later on with some specific examples. With this very short background, I want to dive straight into some of my own research. First, by taking you all once across the world to Western Australia. For 7 years, I was part of the Dolphin Innovation Project, which is a long-term and still ongoing project on a large population of Indo-Pacific bottlenose dolphins in Shark Bay in Western Australia. Since 2007, several hundred, if not thousands, of individuals have been identified, and it's really one of the best studied dolphin populations in the world. And there's very detailed knowledge on their behavior, genetics, habitat use, and also their social system. The dolphins in Shark Bay are characterized by these fission-fusion grouping patterns, meaning that larger groups can split into smaller subgroups of like varying composition, and then they may rejoin or break up again on relatively small temporal scales. Groups of females are usually formed among related individuals that share the same habitat preferences, so they're usually made up of mothers with their offspring over several generations. Dolphin males, they actually build alliances with other males to be able to capture and defend receptive females against others. And these alliances are not based on kinship, but they're usually formed based on age similarity and familiarity as they reach reproductive age. So essentially, those that grew up together are then more likely to form alliances later in life. And these alliances are highly stable over time and often last their entire life. Dolphins in Shark Bay are also known for a wide variety of different foraging strategies, and 2 of those even involve tool use, namely the so-called sponging and shelling. And these behaviors are only shown by a subset of individuals in the population, and they have also been my main focus of the master's and PhD research. During sponging, dolphins wear these conically shaped sponges on their mouth when they forage. So it serves as sort of a protective glove for when they go dig in the sand for prey that is buried. So it protects them from abrasions and also stings from hidden animals. And sponging doesn't just occur anywhere, but is restricted to these deep water channels that you see in the dark color. And the sightings of this sponging behavior actually correlates with the natural occurrence of the basket sponges. Sponging is a commitment. Spongers carry these sponges pretty much all of the time when they're foraging. So it's a very specialized technique. And when looking at fatty acid profiles from blubber samples, it was also found that they have a significantly different diet to those that don't use sponges, since they can target these fish that are specifically hidden under the sand that the non-spongers cannot access. So they really have their own specific foraging niche. And interestingly, sponging is very female biased, with about 80% of spongers being female, and all of those females seem to be maternally related. So in the early 2000s, Krützen et al. Have wondered how such patterns came about, why only some individuals showed the behavior, and what mechanisms were driving the spread. And to address these questions, they used the so-called ethnographic approach, also called method of exclusion, that was very popularly used at the time to essentially identify the differences in behavioral repertoires between groups or populations, and then trying to find the most plausible genetic and environmental factors that could potentially give rise to these observed differences in behavior. And if those factors could be excluded, then it was assumed that the behavioral differences must be due to social learning. So very briefly, in the case of sponging, they first looked at environmental factors by looking at where does sponging occur relative to other foraging behavior. They found that other dolphins also use these same areas for foraging. So it was concluded that it's unlikely that the environment alone is responsible for the behavioral variation observed in this population. Similarly, for genetic inheritance, they considered 10 different patterns of autosomal, X-linked, Y-linked, and mitochondrial DNA-based inheritance, but dismissed all of them since they did not fit the observed patterns. So because environmental factors and genetic factors seem to be insufficient to explain the transmission of sponging, it was concluded that it must be spreading through social learning. And given the mother-offspring similarity in behavior, they concluded that it must be passed on through vertical social learning between mothers and their primarily female offspring. The ethnographic approach, however, has been receiving criticism since, first of all, it's very difficult to consider all possible environmental factors and genetic pathways. And two, perhaps almost more importantly, environmental factors, genes, and social learning can simultaneously drive behavioral variation. So these factors are not necessarily mutually exclusive. So researchers started calling for more quantitative methods that actually help to quantify the importance of social learning, environment, and genes on the spread of behavior. One of these frameworks that was developed was the so-called network-based diffusion analysis, short NBDA. This is a method I use very regularly in my research, and we will encounter it repeatedly during today's talk. So I do want to spend a couple minutes just explaining the basics behind it. Now, NBDA tracks the spread of a behavior through a social network and infers that it is socially learned if the transmission follows the network connections, because it assumes that more closely associated individuals are also more likely to learn from one another. So in the example network on the right, if this bottom individual here came up with something new behavior, found some information, if it's spread entirely through social learning, we'd expect the spread to follow the network, something like this. If instead the behavior was entirely asocially learned, so innovated by each individual, we'd expect the order of learning to be completely independent of the network. So for my PhD, we used an extension of NBDA that includes several networks as a more quantitative way to assess how sponging actually spreads within the population. So to disentangle the relative contributions of these drivers, we used actually 4 different networks. In a first network, as a proxy for the importance of habitat, we just calculated overlaps of individuals' home ranges to see whether similarities in space use influenced the acquisition of sponging. In a second network, we calculated dyadic genetic relatedness among individuals. So as part of the long-term project, individuals are biopsied with these biopsy darts that just remove a little piece of skin and blubber. They can then be used to extract DNA and estimate relatedness. To assess the importance of social learning on the spread of sponging, we actually included 2 different networks that reflect different pathways of transition. First, to assess the importance of vertical social learning, we created a network that only allowed directional learning between mothers and their offspring. And finally, to test for the importance of non-vertical learning, so this includes both horizontal and oblique, we generated a network that reflected the association strength among individuals. So how much time do they normally spend with each other? We found that models that only included this mother-offspring network received most support, but very little evidence for any of the other networks or network combinations. And we also found that females were significantly more likely to learn sponging socially from their mothers compared to males. And there was no influence of matriline membership on the learning rates. So simply being a member of a particular matriline did not predict whether an individual would start using a sponge or not. So what does this all mean? Taken together, these results suggest indeed a strong role of vertical social learning of sponging from mother to primarily female offspring. So while environmental factors are clearly important in determining where sponging can occur in the first place, our results show that both environmental and genetic factors are unimportant for the diffusion of sponging once social learning has been accounted for. And vertical transmission is very much in line with the typical transmission mechanisms for many of these more complex and specialized foraging behaviors that are found not only in dolphins, but in toothed whales in general. And this is unsurprising insofar as these long periods of maternal care of several years in duration, they provide loads of opportunities for learning. And this leads to the typical mother-offspring similarity that is often observed in toothed whales. You might also wonder, why is this strong female sex bias? We don't fully know, but it was suggested that this could be due to differences in these sex-specific reproductive strategies, because males need to focus on establishing these strong social bonds with alliance members to be able to gain access to receptive females. They may not be able to invest time into learning this complex and very difficult to master activity like sponging. And in fact, it has been shown that for spongers, it takes several years before they reach their peak foraging efficiency. Alternatively, it's also possible that this is simply due to the fact that female offspring tend to spend longer in association with their mothers. So the male offspring, they're usually weaned a lot earlier, and that might simply not be enough time to really learn that behavior during dependence on their mothers. Now, sponging is not the only remarkable tool-using behavior that these shark-bait dolphins are capable of, because some individuals have also figured out how to use empty shells of giant gastropods, so sea snails, to catch fish. They would chase a fish into the empty shell and then bring the whole thing up to the water surface, and then they shake it above the surface to drain the water out of the shell until the fish essentially falls into their open mouth. And naturally, we also wondered why only a subset of individuals shows this behavior and what mechanisms were driving its spread. Very similarly, we again employed multi-network NBDA, this time using only 3 networks. A first one, again, using dyadic home range overlaps to test for the importance of habitat. A second one with genetic relatedness to test for the importance of genetic factors. And for social learning pathways, we here only included the association network and no mother-offspring network, because all but 1 dolphin actually started shelling as adults and not as calves. Looking at results, we did again find most support for models that included the association network only, suggesting that shelling is actually transmitted socially among associates. And we found no sex bias in shelling, so both females and males engage in this behavior, and shelling individuals were from 3 different matrilines. And this is further suggestive of a transmission among associates rather than within their maternal lines between mother and their offspring. And so why is shelling different? Unlike these highly specialized techniques like sponging that take years of practice, shelling is a lot more opportunistic. So all of these dolphins that use these shells also mostly engage in other techniques and just every once in a while pick up a shell. And so it might be much more easily learned outside of this close mother-offspring bond. So exposure as adults to associate that use these shells might actually be sufficient to pick up the behavior. Now, why do we even care about how animals learn things? There's some very good reasons. First of all, understanding the capacity for animal learning is important because the modes of transmission determine the efficiency of the spread of adaptive behavior. Take vertical learning, for example. This is a very conservative way of learning that goes across generation. It is slow to change, and animals relying extensively on vertical learning might therefore be at the risk of learning outdated behavior if the environment changes. In contrast, horizontal learning allows for a much more rapid spread of adaptive behavior, and species with the capacity for horizontal learning might be less vulnerable to environmental changes. And second, socially learned behaviors, I guess, can have direct fitness consequences. And I want to illustrate this point again with our dolphins. In 2011, the entire west coast of Australia was hit by this really massive marine heat wave that caused water temperatures to rise by several degrees Celsius for several weeks. And Shark Bay experienced a devastating loss of over 90% of its habitat-forming seagrass beds in certain areas. So all these areas in red. And this, both the water temperature as well as the loss in seagrass caused mass mortalities of fish and invertebrates across all of Shark Bay. And naturally, we wondered about the impacts on species on higher trophic levels, including the dolphins. So what we did is compare demographic data from before and after. First of all, I wanted to look at the graph on the right. We found a sustained reduction in reproductive rates after the heat wave compared to before. So they had fewer calves, as well as a decrease in survival that you can see in the graph on the left. What was interesting, though, is that the survival of sponging dolphins was less severely affected compared to the one of non-sponging dolphins. So it appeared that this socially learned knowledge of sponge tool use allowed them to access a foraging niche that at the bottom of these deep water channels that appeared to be less affected by the heat wave. So being able to use that sponge provided some buffering against the negative effects of that heat wave. So social learning can have direct fitness consequences. With that, I want to leave the dolphins for the time being and move on to my second study organism that I have worked on, namely the European great tit during my postdoc in Germany. Similar to the dolphins, great tits also live in these fission-fusion flocks. But unlike the dolphins, their life history is very fast-paced with a life expectancy of up to about 4 years. And over the next 20 minutes or so, we want to see how their life history and social system might influence their acquisition of behavior as well as decision-making. Now, great tits are a really great study organism for a wide variety of questions because they can be tracked quite easily in the wild using radio frequency identification techniques. So they're caught in these mist nets that you can see on the very left, and they're given a leg ring with a so-called passive integrated transponder or PITAC, so this little gray thing here. And this gets activated whenever it gets into proximity of a receiving antenna. And this allows for remote data collection and helps to generate really high resolution data sets, which have been used to study the very detailed mechanisms under social network dynamics and learning processes. Now, tits live in these mixed species flocks, and it was found that they rely very extensively on social information use in different behavioral contexts, and that the information really does follow the network connections. So the network structure really determines what social information is available to which individuals. Several studies have shown that tits use social information to locate sources of food. And in one of our studies, led by 2 former students, we have also shown that they use social information in a nest building context. So we deployed several dispensers across the study area that we equipped with these RFID antenna, filled them with wool of 2 colors as a source for nest lining material. And we found that the female tits use social information from their foraging associates to locate those dispensers, and also found that the choice of color was likely socially influenced. Great tits are also very capable social learners of behavior, and this has been demonstrated in various cognitive experiments using experimental puzzle boxes. And this particular study, the birds were presented with a puzzle box with a sliding door that you've heard all about, probably during Lucy Aplin's seminar. So they can slide the door either to the left or right to get a food reward. And this study has shown that the solutions to these cognitive puzzles can spread really rapidly through population through horizontal learning. And also that kids tend to be conformist learners, meaning that they show a preference for the behavior that is shown by the majority in the population. Now, this study also built the starting point for one of the studies I was involved in. That was also part of the suggested reading for today's seminar. In this study, we were actually interested whether great kids are also capable of learning a more complex behavior, and if so, what the underlying mechanisms were. This is a multi-step experiment that was conducted by Lucy Appling during her time in Oxford, and we later completed it during my postdoc years working in her lab in Germany. So in the first step of this multi-step experiment, these several subpopulations were exposed to this sliding door puzzle, and they were each seeded with a couple of demonstrator birds that had been trained in captivity. In a second part, birds of the same subpopulation were given access to a different puzzle with a dial that could be turned clockwise or anti-clockwise to get access to a mealworm, again, seeded from a couple of trained demonstrators. Now, in the third part, these two solutions were combined into a complex puzzle where the birds could either produce a one-step solution, so either slide or dial to get a sunflower seed, or then produce a more complex two-step solution by combining the dial and the slide action to get a mealworm, which is the preferred reward. Then finally, in the fourth part, the same puzzle was deployed, but now only rewarding the complete two-step solution. So only producing a slide or a dial didn't get the birds anything. So we were interested in the process underlying the acquisition of both the single and two-step solutions. So are great kids capable of learning a more complex two-step solution? And if so, what are the underlying mechanisms? Now, to address these questions, we actually ran 9 different NBDAs. I'm not going to go into detail on these, so I'll just try and summarize the results. If you've read the paper, you see there's a whole lot in there. So first of all, for both the slide and the dial, we found that the majority of birds who did learn to solve the boxes did so by social learning. And this is not very surprising given what we do know about their capacity for horizontal learning from previous experiments. Now, in the third part is where it gets interesting. Remember the one-step solutions that they still give a reward of a sunflower, and the two-step give a mealworm. So for the one-step solutions, we found that these are still learned socially from others, but that for the complete two-step solution that is rewarded by the mealworm, that this is learned asocially. So what that means is that they learn dial and or slide through copying others, but then they take that knowledge about the single solutions and put those two steps together without any further input from other birds. And we also found that even just knowing slide or dial was usually enough for the birds to asocially get to the full two-step solution, although knowing both definitely helped them to get there faster. In the fourth and final step, where the single solutions were no longer rewarded, it was quite interesting to see that the one-step solutions still were produced, even though they didn't get them anything. And the results were still very consistent with the third part, in that the single-step solution were still learned socially from others, but then reconstructed asocially into the complex solution. And again, knowing one of the solution was usually sufficient to be able to figure out the full complex solution by themselves, but they were faster if they knew both components. So to get back to your questions on whether great kids are capable of learning a two-step solution, the answer is yes-ish. Because even though complex solutions were produced in both part 3 and then part 4, the proportion of birds that could actually do the complex ones declined quite massively from one to the next generation. And completely naive birds were very unlikely to learn the two-step solutions when the single steps were no longer rewarded. So it is safe to assume that probably after another generation or so, the complex behavior would have likely died out. So we're really, this scaffolding of rewarding that first step is very important on the way to learning the complex solution. As for the second question, we have seen that they use a combination of learning strategies, mostly social learning for the single component, and then asocial learning for recombining them into the complex solution. Now, to take this a step further, we were also interested in the consequences of these transmission mechanisms on both the individual decision-making of whether to go left or right, but also what that meant for the emergence of local traditions. So this is showing the numbers and types of solutions that were produced in each subpopulation. So each subpopulation is in the column, and this is just showing the dial and the complex solutions. So if you think about it, for both slide and dial, we only have 2 options each. So for slide, it's either left or right, and for dial, it's clockwise or counterclockwise. But for the 2-step solutions, we actually have 8 possible, depending on whether the birds first start with dial or first start with slide, and also whether they go left, right, or clockwise or counterclockwise. So first, to figure out whether there were any preferences on both an individual and or subpopulation level, we calculated the Shannon entropy, which can give you a measure of predictability in a system. So here we see the preference on the individual level. So a low value means that individuals always choose the same variant, and a high value of 1 would mean that their choices are completely random. So what we can see is that the birds are actually most faithful when it comes to sliding. So they always have their very strong preference for either going left or going right, and then followed by dial, where they're a little less faithful, but still have very strong preferences. And they were even quite consistent in the 2-step solutions that they produced, but we found that they usually stuck to about 2 out of the 8 possible variants, even though some birds produced up to 6 different ones. On the subpopulation level, this is just a complex solution, we found no evidence for a preference of a single solution, but we found that birds still shared a preference for a smaller set out of the 8 possible ones within their subpopulation. So that was more strongly than expected by chance. And this is very much consistent with the NBDA results in that they seem to socially learn the single component, but then recombine them asocially into complex solutions. So they're still kind of sharing a pool of socially learned single variants, but then that might lead to a slightly larger pool of complex variants. Now for the final part of this talk, we are staying with the learning in great tits, but changing in developmental stage, namely from adults to juvenile birds, which is a period that is often understudied in wild animals, and this is definitely also the case for great tits, partly because juvenile birds have high mortality and they're a little bit hard to track after they leave their nest. So great tits are seasonal breeders. In early spring, they establish strong pair bonds within their larger social network. The male establishes a breeding territory within which the female builds a nest in a tree cavity. And then she lays an egg every day until the clutch is complete and incubates the egg for 13 days until the chicks are ready to hatch. Both parents then feed the chicks in the nest until they're ready to fledge, so ready to leave the nest after about 3 weeks. The juvenile birds then continue to be fed by their parents for anything between like 10 to 30 days. So during that time, they roam around in their little family groups until they're ready to start feeding themselves and reach full independence. So while this period from hatching to independence is quite short, there is this time of really increased social tolerance within that family unit, both between parents and their offspring, but then also among siblings of the same clutch. So naturally, I was curious as to how this short but intense time of being in the family unit would influence the learning strategies that these juveniles use during transition to independence? Who do they choose as role models, and how do these strategies change as they get older? To get at these questions, we conducted, again, an experiment in southern Germany where we had 2 breeding populations where we deployed about 300 nest boxes. We monitored all breeding activities with equipping adults and chicks with pit tags, and then deployed a grid of feeders across the study area equipped with these antennae. And this allowed us to keep track of how the associations within the family units as well as outside changed during transition to independence. And we have a study out very recent that details a lot of these dynamic changes in sociality during that time, if you want to check it out, out like a month ago in behavioral ecology. This is important because the social network essentially determines what information is available to which juvenile at which point in time. So we needed to keep track to know what behaviors they could possibly learn. To look at the learning strategies, we conducted another puzzle box experiment based on the sliding door puzzle we've already encountered. But we made the whole thing selective so that we could control which side the birds had access to depending on their individual pit tag. So as the bird landed on the antenna on the perch, the box selectively activated one or both of 2 little motors on the inside of the box to unblock the door to one or both sides. And this allowed us to create 4 different experimental conditions within the family units. First, we had breeding pairs that both knew how to solve the box. We forced about half of them to solve on the same side and the other half on opposite sides. For breeding pairs with only one solver, we just restricted that one to either left or right. And then we had controls of breeding pairs where none of them were able to solve the box. Now, there's an important thing to remember because solving is not the only way to get access to a mealworm. Because as an alternative solution, individuals can also scrounge by very quickly flying in after another bird has solved to also pick out their worm before the door closes. So really, there are 3 different strategies. Now, we let this experiment run for several weeks, recording all solving and scrounging activities by adults and juveniles, including when each bird started to solve, and in the case of solving, which side choices they made across time. And the juveniles participated incredibly eagerly in the experiment. In only a few weeks, we went through over 30 kilos of mealworms, so that was very intense. And out of the over 100 juveniles that survived in independence, actually 53 learned to solve and 75 learned to scrounge. And together they produced over 10,000 solves and 3,000 scrounges. And this really rich data set allowed us to really ask various questions on the learning and the underlying mechanism. So first of all, we were interested in the role of parents in predicting the strategy that offsprings employed. And we found that the parental strategy was actually quite a strong predictor of what strategy the offspring then used. So on the x-axis, you see the number of solves that the parents produced during dependence. And on the y-axis, it's the number of parental scrounges. And the offspring strategies are shown in different shapes. So if the parents neither solved nor scrounged, so at the 0-0 crossing, then the offspring were unlikely to show any of the behaviors themselves. So all these little crosses down in the left corner. Then moving up on the y-axis, those with high frequency scrounging parents, but few solves, these offspring were most likely to acquire only scrounging, so all the triangles. And as the number of parental solves increased, juveniles were most likely to learn both strategies, which are shown in circles, so both solving and scrounging. So clearly the parents had something to do with it. But next, we really wanted to tease apart who the role models were during ontogeny. And we did this again in a framework of 2 separate multi-network NBDAs. You know the drill by now, one for solving and one for scrounging, where we use different networks that reflect different pathways. What we did here is that we subset the association network that we collected weekly into subnetworks that allowed for different pathways of learning. In a first network, we tested for the importance of parents as role models by only allowing connections between parents and offspring. A second network where we only considered associations among non-parental adults and juveniles to test for oblique learning. A third network where we only had connections among siblings to measure the importance of horizontal learning, but specifically within the family unit. And finally, another horizontal network, but this time only with the connections among non-siblings. And again, we modeled the diffusion of both scrounging and solving along all combinations of the different pathways, using the time at which the juveniles learned as diffusion data in the NBDA models. As for results, overall, we found that both for solving, that's on the left, and scrounging on the right, we found very strong support for social models over asocial models. And those social models that included the parent-offspring network, the one between juveniles and non-parental adults, and the one among siblings, but not the one among non-siblings. So essentially a combination of vertical, oblique, and horizontal learning among siblings. Now, interestingly, when looking at the effect sizes, it seemed that for both solving and scrounging, the learning was strongest among siblings, followed then by transmission between non-parental adults and only then by parents. And we'll come back to what this all could mean in a minute. Because we took this a step further to distinguish the typical pathways within the sibling cohorts. So we distinguished between first learners between a sibling cohort that are shown in white and the subsequent learners that are shown in gray. So what we can see is that the first siblings for both solving and scrounging tended to learn either through vertical or oblique learning, but then that the subsequent learners mostly relied on their already knowledgeable siblings for acquiring both of the behaviors. Now, as a final piece of the puzzle before we look at the big picture, we wanted to know a little bit more about what exactly these juveniles learned from the others by looking at the side choices of solving. So now only considering the solves and not the scrounges. So how do the juveniles decide whether to push the door left or right? And how do these strategies change over time? We looked at this decision-making at different points in time. First of all, we only looked at the very first solving day when they were entirely inexperienced. We looked at whether the juveniles paid any attention to the parents' solving behavior during dependence. But we found no evidence that they were following their parents' side choices, and this effect remained regardless of whether the juveniles had received conflicting information or not from their parents regarding the side choices. Instead, we found that juveniles on their very first solving day were strongly influenced by their social environment, meaning that the solves that they observed immediately prior to making a decision influenced whether they would go right or left themselves. So if the majority of birds were solving right, they were more likely to go right themselves. If the majority went left, they were more likely to solve left themselves. And this was the first solving day, but what about when the juveniles started gaining more experience? So in this model, we now looked at all the 10,000 solves and how the reliance on social information changed with increasing experience. So here the different shades of blue indicate different solving days, with darker shades meaning more experience. And what we can see is that the more experience they gained, the less reliant they were on social information, shown by the decreasing slope. Instead, the overall side choice was predicted by personal experience. So it seemed that once they figured out how to solve one side, they usually stuck with it. This was a lot of puzzle pieces, so we want to take a minute to put this back into context. First of all, we've seen that the parental strategy, so whether they solved and or scrounged, was predictive of the strategies that the offspring are showing, indicating that early exposure does facilitate learning of particular behavior. When we look at the NBDA results, though, we find that there's most support for transmission from siblings, followed by learning from non-parental adults and only then by parents. And this indicates that the actual acquisition of the behavior only occurs after they have largely separated from their parents. And this is nicely supported by some of the video footage. I'm just going to make this quiet. So these are a bunch of independent juveniles. They're meant to feed themselves by now, but they clearly have absolutely no clue on how to solve the box just yet. And this really supports our results that learning to solve the puzzle only occurs after they reach independence. And it was also very visible that these juveniles do really use each other as potential sources of social information. They follow each other's actions very closely. If one starts pecking and pulling on a little piece of tapes, the other will immediately gather around to see what's going on. Now, for site choices, we found that initially juveniles resort to using social information, that with increasing experience, they largely rely on personal over social information. And this is consistent with a strategy to learn socially when you're uncertain about the decision. But as you get better, you can rely on your own experience. So overall, in great hits, learning from parents does occur to an extent in that the offspring do adopt their parents' foraging niche, and early exposure facilitates learning. But that for acquiring these little more complex behaviors such as solving or scrounging, the dependence period might actually just be too short, and that therefore the juveniles then need to rely on other non-parental sources of information instead to really get to the full behavior. Now with that, I want to circle back around and put what we have learned into the framework of my research interest that I have shown at the very start. First of all, we have looked at the tool tweezing behaviors, sponging and shelling in the dolphins that are shown by a subset of individuals in the population. We have seen that these behaviors are socially transmitted, sponging through vertical social transmission from mother to mostly female offspring, and shelling non-vertically among close associates. We've also seen that the cultural repertoire that these dolphins have, so these socially learned behaviors, have direct fitness consequences because spongers had better survival after a marine heat wave compared to those that do not know how to use sponges to forage. We have then looked at one of the puzzle box experiments in adult great hits to see whether birds are capable of learning a more complex two-step solution. We have found that the single steps, slide and dial, did spread through social learning, but that for the two-step solutions, birds used asocial recombination without any further social input. For the decision-making on the individual level, we have seen that the birds were quite faithful to their preferred one- and two-step solutions, and that on the level of the subpopulations, these learning and decision-making patterns led to the emergence of shared preference for a subset of all possible solutions, which are, I guess, somewhat consistent with local traditions. Finally, we have looked at the ontogeny of learning strategies in the graded juveniles using this sliding door puzzle. And we have seen that during ontogeny, the juveniles learn these behaviors socially from others, with siblings being a very important source of information, followed by non-parental adults and parents, even though early exposure facilitates learning. For decision-making, so whether to go left or right, we have seen that initially they rely strongly on social information, but that with increasing experience, they rely more on personal information. Now with that, I really want to thank my collaborators, advisors, and of course the funders who have made this work possible. And all of you for listening, and also, of course, for the invite to give today's seminar. It was my pleasure. Thank you.
Thank you very much, Sonja, for such a great talk. So we now open our discussion time. Please, as I set up in the chat, please state a question mark for your question or an S if you're a student or an early career. We'll give you priority to everyone. We will give priority to students and postdocs. And then whenever you ask your question, please state your name and affiliation. That will help us, create community. And I think that's everything. So I'll put you up. Eduardo, could you please help me checking out the YouTube?
Yes.
For questions? Yeah. Okay. We've got the first question from Eduardo.
Thank you. Eduardo Fernandez Duque, Yale University in the United States. Sonja, thank you so much. I feel like I know you already. So one of the things that I really appreciated, and it speaks so beautifully of some of us have chosen a path of focusing on one taxon and one system for a long time. And you've done wonderful work cutting across 3 different taxa. I mean, water, land, you name it, birds, mammals, and really bringing a lot of integration to some central ideas on learning. So thank you so much for that. And I hope to come back with some questions later if there's time on the specifics of that. But I just wanted to get out of the way a very pragmatic one. Can you tell me very briefly about the bio darts? Of course, you're recovering them. How big are they? What is the company that makes them? Very pragmatic. Just give me the basics or the person that I can contact. Has it worked well? How big is the punch you're getting from the dolphins or you are getting from the dolphins?
Absolutely. I cannot, no, I do remember the company just now. I think it's Pax Arms. And there is actually a paper by Michael Krützen from the early 2000s that kind of details this. The darts are about, they exist in different sizes. We have pretty much the smallest size, I guess, because our dolphins are small compared to like a large whale. They do work either with air guns that we use. They can also be used with what are they called? Oh my God. I forget the name of the.
Don't worry. But
Yeah.
I mean, do you remember how big? It's not as much
I
the
wanna
size of the dart
say-
as it is the size of the sample you're getting.
Absolutely.
Because if you think dolphins are small, wait to see owl monkeys. So
Yes.
do you remember how big they are, the punches that you were getting?
Yeah, it depends a little bit on where exactly you hit by the body, but if you go slightly below the fin, that's where it's best. Usually takes out between like 5 millimeter to 1 centimeter long bit. So it is really, really plenty of material for loads of DNA analyses. And even if like, the angle isn't perfect, it usually gets out enough of the skin to get good DNA. At least we've done mostly like genotyping with it. I know that the group also does a lot more genomic work, of which obviously you need a little more material, but this has worked really well for us.
I'll check Michael's paper. Thank you so much.
No worries.
Thank you, Eduardo. And then we have a question from Michael.
Yes, thank you very much. Michael Taborski, University of Bern. I wonder to which degree you are interested also in the type of learning mechanisms that is responsible for these different traits that you find and study. Because, I mean, there is, of course, different complexities regarding the cognitive requirements for these different learning mechanisms. And I could imagine that a lot of the social learning and accomplishment by social interactions is on a relatively simple basis, like, for example, social facilitation or local enhancement, and not really copying or emulation independence of local enhancement, for example. So I wonder to which degree you're interested in these different learning mechanisms and what information you can provide about involved learning mechanisms in the study systems that you presented?
Yeah, very, very excellent question. I want to say I'm interested in it. I feel like that my studies have not quite provided really enough data or like detailed data to really make statements about what learning mechanisms are at play. For example, with the dolphins, it's hard there it's kind of hard to it's very hard to really say anything because most of the behavior actually happens underwater. So what we see is when they surface with the sponge or the shell and when they come up for breathing, so we don't actually know what happens at the bottom. So we don't really know if there's like variation in how you use the sponge or what sponges exactly that you use, or we know there's size differences, but so far we haven't really seen a pattern that some dolphins always use a small one and others always use a big one, and how that relates between mothers and offspring. As for the tits, I guess there we have a little bit more of an idea. Given that they do actually copy the site choices, I feel like the mechanisms must be a little more than just like local enhancements or like facilitation. So I guess there is some copying of the actual like motor pattern involved, but to what extent that could be like imitation or something else, I wouldn't like necessarily dare to say that we do have the answer. It's clear that for the two-step task, they're clearly not copying. It's more of a like, okay, they have like we just discussed this in the paper a little bit as well, like why do we see this low social learning for the one-step solutions, but not for the two steps? And one argument that we make is that there might be some cognitive limitations in that they're not able cognitively to really pay like enough attention to both observe and then produce twice as much information. So interested, yes, but I don't know if we have enough to really say what's happening.
But could it be also in the one-step solution that this is a combination between social learning by, for example, local enhancement, and then individual learning by just trial and error? And because of the fact that they are interested in the particular place where it happens, then they try out left or right and find one of the two solutions more successful, and that is actually then the learning mechanism that helps them to solve the problem more reliably in the future.
It is a possibility, and I think like local enhancement is a very strong one. Like even the juveniles, I learned extremely quickly that the box, like the surrounding, meant food somehow. So there's clearly a strong attraction to the site. What I think speaks against the asocial learning part is that we do see this like really strong conformist tendency. So I feel like there's some information about the direction of this movement of the door is being transferred. So this would speak against like, oh, they're just attracted to that front of the puzzle box, but more for like, oh, they're actually observing some of those motor patterns that are being produced.
So the conformity is already with the first move that they make.
Yeah,
okay.
Yeah, it seems like it, yeah, from the very start.
Okay, thank you very much.
Of course, thank you.
Thank you, Michael. Our next question comes from a student, Julian.
Hello, everybody. I'm Julian. I come from Rosario University in Bogota. Sonia, I would like to know more about the male dolphins that have the sponge pharyngid strategy. Do they know from juvenile stage have this type of strategy, or it has been an accurate learning from the conspecific? It's like been switching the strategies from pharyngid?
Can you just repeat one part? I like do they do it from the very start, from very early? Is that ontogeny wise, where you said, is that what you're asking? If they're doing from the very start, or if they're switching?
Yeah, that is the two-part
Okay.
question.
Yep. Okay. So yes, they do it actually from really early on. We see usually like the calves that are born to sponging mothers, they start picking up a sponge when they're about 2 years old. But I guess more in a playful way because they're still being nursed by their mother, so they don't really need to forage just yet. And once they're independent, they're already sponging pretty much 100% of the time. So they usually, if you committed to the life of a sponger, you do so from very early on. And there's not usually a any switching in strategy after that. There's other behaviors that are kind of like more generalist, but sponging is really like an early on commitment and you stick with it your entire life.
Do you have any idea is that correlated with like an imprinting thing, or is just a vertical learning?
We think it does not necessarily, by imprinting you mean genetically?
Yeah.
Yeah, we think that imprinting is unlikely because there is the genetic patterns do not quite fit. So there are calves that are born to sponging mothers, mostly males, that do not pick up the sponging, but it still happens. So still about 20% of the male offspring will pick up sponging. So we're thinking that it's really like a question of exposure, that the time of dependence is really determinant of whether they do learn the sponging or not, and less so a genetic imprinting.
Okay, thank you.
Thank you, Julian. And then we are in a row with the students. We have Louis.
Yes. So, hello, I'm Luis Darie. I'm a student 2 in Colombia in the University of Rosario. First, thank you for the presentation, but I had a question. I don't know, it's not really purely related with your first 2 studies, but I was wondering how groups that express I don't know how to say it, but hierarchical, or like with a hierarchy structures, how do they express social learning? Like, is it mostly influenced by this here hierarchy or not?
There is a bunch of studies that have looked at that. I couldn't like name you any particular ones right now, but there's definitely social learning strategies that are influenced by rank, with sometimes role models being specifically chosen as the ones who have high ranks. It's probably in the primate world that you will find examples of this, where they're really specifically copying those like prestigious individuals that had hold high ranks. But I couldn't tell you if that was like a general rule that applies to most animal species with that are hierarchical.
Okay. Thank you. And for example, in squirrels, it's also done by family on what you find.
I don't know yet for the squirrels.
Okay.
I'm currently like, you can't see because my background is blurred, but there's a whole mess of puzzle boxes being built behind me right now. I'm about to start field work. And my theory is that there will be some influence of like maternal transmission, because the pups, after they do leave the burrows, I work on ground squirrels, so they're, emerge from the burrows at some point. They do spend some time with their mothers during that first time, which could be a potential time to learn, and we do see them interact quite frequently during that time. But I will be able to tell you hopefully in a few months.
Okay. Thank you.
Thank you, Louis. And then we have a question from Natalia. Natalia, are you there? Natalia Cunha. We'll give her like a second. Maybe she'll pick up, or I will talk to her in the chat. And meanwhile, we can go with Susan.
Hi, I was just wondering if you could say a tiny bit about your future research involving personality and social learning in the ground squirrels. I'm doing something like that in capuchins right now, and just wondered how much our interests overlap. Clearly, we should be talking more often. This has been a fascinating topic.
Awesome. Yeah, that sounds great. Yeah, sure. So in a nutshell, I'm interested in how the presence of humans affects social networks and learning strategies. I work with Jen Smith on her long-term project here in California, and we do have these two populations that differ in their level of disturbance. So one is like very pristine. There's an occasional hiker that goes through, but they're quite reactive to humans, so they're quite shy. The other ones, like next to a parking lot, there's picnic tables. They're very used to people and have very high human tolerance. And there has been some work looking at how disturbance by humans affect their network connections, and it's shown that on a short term, it disrupts their connections. So I'm interested in kind of like how these disturbance environments as like a more like environmental, like a longer term effect affects their network structure. So I'm using a lot of like RFID techniques to kind of map their co-foraging. I'm mapping like what are the disturbance levels during the day while we do this data collection. And then also how does it affect their learning strategies, with the idea that in the disturbed population, because they do have like reduced connections, that they would be relying a lot more on asocial learning. And the personality part comes in where we're doing a lot of flight initiation distance trials to see how human tolerant are they. And I'm hoping to build that in as sort of a component into all of my analysis, like, yes, their network is disrupted, but this is really like has an interactive effect with how human tolerant they are. And same with the learning, are the ones that are more human tolerant better social learners because they're not faced so much by the human presence. So these are kind of like the questions I'm looking at the moment.
Okay, so the flight distance is sort of like a Shy-Bold assay.
Absolutely.
Okay.
Yeah, exactly. And it works quite well and has a huge variation. First of all, it has a huge variation between squirrels, but it's quite consistent within squirrels. So we have some really good data on that. This is quite a good assessment of how human tolerant they are.
Wonderful. Okay, thank you.
Thank you, Susan. And now we can have, it's not Natalia. That's why she didn't pay attention. So it's Valentina. Valentina, you can answer your question, and then we give the word to Sule.
Hi, yes, I have a problem because I am in computer of my university and the username changed. But I'm Valentina. I'm a student of Universidad Rosario in Colombia, and thanks for your talk. It was very interesting. And I have two questions. First is in species that have a group care of their offspring, how does vertical learning occur?
Sorry, the acoustics are a little bit bad. In species with what?
Group care of offspring.
Care. Yep. Yep. Good question. There is a really cool paper on mongooses, I believe, is it? Where they actually have other adults caring for the offspring rather than the parents, and there's very clear similarities between behavior between the non-parent adults, between the carers and the offspring. So this is a really beautiful example of like, social proximity does actually influence like the transmission patterns. And this is definitely something that I'm hugely interested in like how does the social system really determine what forms of generational learning these animals rely on. So obviously like a lot, like in species with long periods of parental care, we do see a lot of vertical learning. But what if that's not the case? What happens then? Where do the juveniles get their information from? Because they usually do not know much about the world. They do need their social sources of information. So yeah, I definitely recommend checking out that paper. I forgot the authors and the title, but if you look, there will be something about the carers in like I believe it's mongooses.
Okay, perfect. Thank you. And the second question is, you talk about the difference in social systems in male and female dolphins. What are the reasons?
What are the reasons?
Yes.
Or so this has a lot to do with their life history. They're very long-lived. They have very long periods between pregnancies. So the young usually stay with their mothers for about 3 to 8 years. So fertile females, or like females in estrus that are ready to mate, are really, really hard to come by. So no one really knows how the alliance formation in the males really started off, but it is now really interesting that without being part of an alliance, a male will not reproduce. It will not stand a chance to be really like access to females. And it is a little bit hard for the males to predict when exactly the female will be receptive. So they usually know the female starting to be in estrus. And how it works is that the males will literally capture a receptive female, and then they will guard her for up to like 2 months or even longer. They will all mate with her repeatedly to make sure that one of the alliance members is the biological father of the offspring. And it's a very interesting system because they're not related like any more than expected by chance. So it's an incredibly interesting system that this is maintained and has evolved in the first place. So these are kind of like, yeah, it's a mind-blowing kind of system where they have very, very different reproductive strategies. And during that time, it's usually you see a lot of like fighting too. There will be other alliances that come in and try to steal the female, and then they will team up with second order allies that come and help in to defend against the intruders. So it's highly political and very, very cool to observe.
Thank you, Sonja.
Thank you, Valentina. Now a question from Zuleima.
Yes, hi. Zuleima Tang Martinez. I'm retired from the University of Missouri in St. Louis in the United States. And you might have talked about this, Sonja. I had to deal with somebody who came to the door. And so when you were talking about the dolphins, I missed some. But I was really struck by the difference in that the sponging seems to be vertical while the shelling seems to be horizontal. And I was wondering if why you think that is. And again, you might have talked about this. Does it have to do with the social context, the types of groups that they're in, or what? And in general, I guess, are there any hypotheses, while even within the same species, some behaviors seem to be learned vertically, other behaviors are horizontal or oblique?
Definitely, yes. I've touched on it very briefly, but it's great that you're asking because there's more things I can say about it. So one part is that we suggest that it has to do with the opportunistic nature of shelling. Because sponging is a commitment, is hard to learn. It takes years of practice until you get better at it. And shelling is kind of a thing they do on the side. So all the shelling dolphins also mostly engage in other strategies, and only occasionally when they happen upon a shell, they will pick it up. And it seems that it's a lot less complex to just, give it a go. So exposure as adults to one of your associates doing it might be sufficient to for you to just try. And there's also a little bit that toothways in general are, they are conservative learners. They're usually like trust mom most. If you as an offspring are alive in the marine world, it means that your mother has done something right. So they're very much reliant on their mothers as role models for anything that matters. So anything like a commitment like sponging, where it's really like, this is your daily bread. You want to make sure that you're following the right role model. As for shelling, there's just a lot less risk involved if you just kind of copy this behavior from someone else, because it's just this very opportunistic thing. And we see it with a lot of like play behavior, for example. They do copy horizontally from other youngsters as well, but there's so little risk involved. And I feel like this is kind of a general pattern that one can see, that for the behaviors that really matter, they're very selective in who they choose as their role models. But the ones that are kind of like, might give it a go, it's not so bad if it doesn't work, then they're a little less selective in who do they choose as social role models.
Okay, great. And is there a sex difference in the shelling, like females versus males? Does one sex do it more than the other?
There was not. So they really both engage in it indiscriminately. There is a little bit of a geographic restriction because, and we think this has to do with where the shells actually occur. We have this very distinct topography in the bay where these like flat kind of, only like up to 5 meters deep seagrass beds are interspersed with these deep channels. And the gastropods, so the sea snails, they live on these like sand flats. And apart from maybe like one time, this is where we see the behavior. And this is so different to the sponging. The sponging occurs only in these deep water channels. So you can kind of, depending on what you want to see each day, you kind of know where to go.
Yeah. Well, thank you so much. It was really a fascinating talk. I really enjoyed it. Yeah.
Thank you.
Thanks.
Thank you, Zulema. Eduardo, you want to go with your question?
Thank you. Sonja, can you remind me, there was so exciting, so much information early in the talk. It was probably the first third or so. You were sharing with us the genetic evaluation of the potential connections between, let's call it, genes and learning. And if I remember well, there was some kind of summary slide where your colleagues had concluded that there wasn't really genetic evidence for this. Now, my understanding is that we're still in such a situation of extremely limited knowledge about behavioral genetics for any taxon, and I imagine that dolphins must not be one for which we know a lot about behavioral genetics. Can you go back again over that? Tell me, how was it that they made a decision to say no? Because I don't remember the proxies they used, but they were saying like, no, we see no connection between X and Y, but we really have no clue about the process by which X being the genes and Y being the learning occur. So I was a little bit lost, confused by how come they can reach this conclusion given how little we know about behavioral genetics.
Yeah, that's a very fair question. And yes, we are in its infancy, right? So what they did is that they essentially they took like the example of like mitochondrial DNA. If sponging was somehow encoded on the mitochondrial DNA, what is the pattern we would be observing? And for that example, they were like, oh, if it's on the mitochondrial DNA, we would be expecting a very strict mother to offspring inheritance of the behavior, but to both males and females. And they were like, well, it's mostly females that do it. males very rarely. So this seems implausible. So this is kind of, and then it went through all, like, I think 10 different pathways of like, oh, if it was on the X chromosome, what's the pattern we would be expecting, and does it fit what we see? But there has been criticism about these approaches because, as you say, it's almost impossible to look at all the different genes or like even like epigenetics that might be responsible for some of the expression of these behaviors. So that's why we're now like coming away a little more from this ethnographic approach by excluding things, but more treating it as like these drivers can act simultaneously. And I think sponging is a really great example of that, where there's clearly environmental factors that interact with the social learning. The roles of genes is still not entirely, like my analysis have shown it's not like we have been genotyping, so we're not done any like genomic approaches. So it's just kind of a crude approach, I guess. And it shows that there's no correlation between any of the genetic inheritance once social learning has been accounted for, but it doesn't mean it's not there. Like there might still be some underlying genetic factors that might be playing into this that we just don't know yet.
Thanks. I think it helps me understand. Just one more follow-up question to make sure I understand. So what I find problematic, and I think you're agreeing with it, and apparently the readership is agreeing with that, is that they're making a humongous assumption, which is that there's a very, very strong genetic determinism for which we're going to find a signature looking at mitochondrial DNA. And so you start with an assumption that most of us will feel is unwarranted, given the state of knowledge of behavioral genetics, and given an assumption that most of us would challenge, then they move forward with this analysis. Am I understanding correctly?
Yeah, absolutely. I need to say that, like, you know, this study was done in, I think, 2005. So we are at a different state of knowledge these days, for sure. And like I think it was a great, like, you know, opening the field to animal culture, which was at the time was a little like, whoa, what, animals have culture and then socially learn things. So I think it was a really great like way to open up like the possibilities. But there have been a lot of papers back and forth about this one, actually arguing what the weaknesses are of this approach, and like, oh, we cannot conclude that that is really happening. So just really opened up a really interesting debate. And I agree with you. Yes, the approaches are nowadays not considered sufficient to really, like, you know, to really say stuff about, oh yeah, this is probably the pattern that we are observing. But I think
I'm thinking 2005. That's fantastic that you share the historical context, because 2005, that's only a few years after the sequencing of the whole genome, human genome.
Absolutely,
so
yeah.
Many in the audience are very young. But right, I mean, if you go back, if you're traveling time, and I think thank you for bringing that time frame, because it was a time when there was a lot of talking and thinking that, okay, now, I mean, we can go back to the genome because we can sequence it fully, and that will tell us whether they are learning because of the genes or not. So thank you. I think it's fascinating to have the historical perspective of how we were looking at and where we thought the answers were and the money was, right? So
Absolutely.
So we just needed to do whole genome in the early 2000s. That's what was going to give us all the answers that we couldn't get otherwise. Thank you.
Absolutely. Yeah. And this is usually, I find it interesting to give that historical perspective as well, because it leads very nicely into development of like of these new statistical methods and frameworks that come with more data. We need something to be able to analyze these patterns. So it's really like hand in hand with like the development of this network-based diffusion analysis that really allows us to look at this more and more data. And I know that the group in Shark Bay, they are working a lot more on like epigenetics, genomics. So there's a lot more being done and being produced. So I'm really excited to see what they do find in terms of like genetic like or genomic correlations with behavior. So there will be more, I'm sure.
Thank you, Eduardo. And I don't see any other questions, so I'm gonna ask mine. Indeed, I do have two questions. First, I want to say thank you, Sonja, for such a great presentation and talk. My questions are related to the two systems that you presented on. So I'm going to ask the dolphin question first. Kind of struck by the fact that pretty much the males don't learn too much the sponging strategy. But you mentioned that 20% of them do, right? And also you mentioned in your presentation that there might be a time constraint because they don't spend that much time with mom or in the group, and they have to leave. So I was wondering if these 20% of the males, they actually spend more time in the group, or what is that is in there that makes them learn versus the other ones that don't learn anything? Is there any particular trait? Is it about personality? Is it about opportunities? Yeah,
it's a good question. Yeah, that's a good question. We don't know because all of the male spongers that we see, or maybe other than like 2, they're all already like 30 years old. So we don't know what their childhood was like, how much time they spent with their mothers. But it was incredibly interesting is that what I didn't mention actually during the talk is that being a sponger also restricts you spatially because you need to forage in these deep water channels and you cannot really go anywhere else to forage. And a lot of males, they roam really, really widely when they go look for females. So it's not really compatible with this, alliance behavior of like going around and trying to find the females. But it's very interesting to see is that the males that do use sponges, they have formed an alliance. So the ones that share a foraging strategy tend to be alliance members. So we really have these like 2 or 3 sponging alliances of the males that then will sometimes capture a female on the flats and bring her back to the channel because that's where they live and forage. So even if she's not really adapted to this environment, she just is like forced to go live in this area. So there's clearly something going on with like trade-offs of like, oh, this is my foraging behavior, but also I need to build alliances to be able to reproduce. So how about I choose the one males that are most similar in behavior to me and has the most like the most similar like needs in terms of time budgets and geographic location. So we don't know what it is, what makes them spongers, but we do have some insights into what happens once they are spongers. How do they make it work, essentially?
That's pretty cool. So they have some sort of associative association based on the foraging strategy.
Absolutely.
Yeah. And if they take a female and the female doesn't sponge, then will she learn that later on in her life, or do you know?
No, she will not learn it. She will try and do the best of a bad job. Like it's not her environment. It works vice versa too. Sometimes you will find a sponging female on the flats, where I don't know how much food she will be getting. Then this is often the case then where the female tries to escape because clearly this is not working out for her. But the males are very good at like forcing her to stay with them. There's usually one male will always stay very close to the female. The other 2 might go off and forage a little bit. But if the female tries to do any escape attempts, the other 2 come in and kind of like, keep her with them. They also have these vocalizations that they produce. They have these like popping sounds that are meant as threats. So as soon as she makes any attempt to like swim away, they will produce these pop sounds, and then she'll usually come back because she does not want to risk any like injuries or a beating or so it's a very like coercive system.
Interesting. And so the second one is about the teeth, so it's going to be a shift in there.
Sure.
So you show that they learn a lot from siblings, but I was wondering, and this is, they are seasonal, I understand siblings will be about the same age, but then would you probably infer if they are learning from older siblings, then I mean, like the youngest from the clutch will learn better from the oldest one, or from a a sibling that it's like more similar age to them?
I think age is not really the determinant, because they're all the same age, right? They will hatch on the same day. They usually fledge on the same day. So they're developmentally, they're all at the same point. And I think that's also what makes the difference. They have very similar developmental needs at the same time. So they seem to be really good sources for social information. And I don't know what determines who the first learners are within a sibling cohort versus the subsequent learners. I had looked at like fledged weight. We do know how heavy they are in the nest, but that does not seem to be a good predictor of like who learns first and who goes later. It is also to say that there's really, really large mortality. So usually, you know, most siblings of like a clutch of 5 that may have left a nest, there might only be 1 or 2 left, but we do have some where we have like 5 or 6 or 7 siblings. So it is quite interesting to see. But I don't know exactly what determines who the first ones are and who the next ones are. But it's definitely, it's not age, it can't be age. They're all kind of at the same level. And I think that's also the reason why we don't see any horizontal learning, or not so much horizontal learning with non-siblings, because they're all kind of at like a different developmental stage. Even a few days make a huge difference at that age. So I think that's why siblings are really important. And in the paper we just published on like the social strategies during that time, it becomes really important, apparent that they start off with the siblings being the preferred associates, and then at some point this comes together and they don't distinguish anymore with other birds. But really, like in these first few weeks, even after they're independent from their parents, they still hang around with their siblings a lot. And only a few weeks later, they're like, oh, we're good now. We have like integrated into the local flocks. We have other friends now. So it seems to be very, very important, having those siblings around.
That's pretty cool. Importance of family. Okay. Thank you, Sonja. I don't see any other question unless someone wants to ask. Otherwise, I'll stop the live stream. Okay, so thank you everyone for coming. Thank you, Sonja, for a great talk and for this discussion that was very enriching. And I just want to remind everyone, we're going to be back on the 3rd of September. Just keep an eye on your emails. We're going to send out the program for the fall season. We'll be celebrating our 4th year. And so just keep an eye on that. Thank you very much. And now I'm going to close this.